Hello everyone, I am Deepanshi from BSc Chemistry Honours final year, roll number 76. Today's topic is biomolecules, carbohydrates. Carbohydrates occur in every living organism. The sugar and the starch in food and the cellulose in wood, paper, and cotton are nearly pure carbohydrates. Modified carbohydrates form part of the coating around living cells. Other carbohydrates are part of the nucleic acids that carry out genetic information, and still other are used as medicines. The word carbohydrate derives historically from the fact that glucose, the first simple carbohydrate to be obtained pure, has the molecular formula C6H12O6 and has originally thought to be hydrate of carbon C6H12 H2O whole 6N. This view was soon abandoned by the name persisted. Today, the term carbohydrates is used to refer loosely to the broad class of polyhydroxylated aldehydes and ketones, commonly called sugars. Glucose, also known as dextrose in medical work, in the most familiar example. Carbohydrates are synthesized by green plants during photosynthesis, a complex process in which sunlight provides the energy to convert CO2 and H2O into glucose plus oxygen. Many molecules of glucose are then chemically linked for storage by the plant in the form of either glucose or starch. It has been estimated that more than 50% of the dry weight of the earth biomass and plants and animals consist of polymers. When eaten and metabolized, carbohydrates then provide animals with a source of readily available energy. Thus, carbohydrates act as the chemical intermediaries by which solar energy is stored and used to support life. C6H2CO 6CO2 plus 6H2O in the presence of sunlight gives 6O2 plus C6H12O6 cellulose or starch. Because humans are the most, other mammals lack the enzymes needed for digestion of cellulose. They require starch as their dietary source of carbohydrates. Grazing animals such as cows, however, have have microorganisms in their first stomach that are able to digest cellulose. The energy stored in cellulose is thus moved up the biological food chain. When these ruminants animals eat grass and are themselves used for food. Classification of carbohydrates. Carbohydrates are generally classed as either simple or complex. Simple sugars or monosaccharides are carbohydrates like glucose or fructose that can't be converted into simpler sugar by hydrolysis. Complex carbohydrates are made of two or more simple sugars linked by acetal bond. Sucrose, for example, is made up of one glucose linked to the fructose. Similarly, cellulose is made up of several thousand glucose units linked together. Enzymes catalyzed hydrolysis of a complex carbohydrate breaks it down into the constituent monosaccharides. Sucrose gives one glucose plus one fructose. Cellulose gives up to 3000 glucose. Monosaccharides are further classified as either aldose or ketose. The "ose" suffix designates a carbohydrates, and the aldo and keto prefix identify the kind of carbonyl group in the molecule. Whether aldehydes or ketone, the number of carbon in the monosaccharide is indicated by the appropriate number called prefix, numerical prefix, tri, tet, pent, hex, or so forth in the name. Putting it all together, glucose is an aldohexose, a 6 carbon aldehydo sugar. Fructose is a ketohexose, a 6 carbon keto sugar. Ribose is a aldopentose, a 5 carbon aldehydo sugar. And pseudoheptulose is a ketoheptose, a 7 carbon keto sugar. Most of the common simple sugars are either pentose or hexose. Depicting carbohydrates stereochemistry, Fisher's projections. Because carbohydrates usually have numerous clarity chirality centers, it was recognized long ago that a quick method of representing their stereochemistry is needed. In 1891, the German chemist Emil Fischer suggested a method based on the projection of a tetrahedral carbon atom onto a flat surface. These Fischer projections were soon adopted and are now a common means of representing stereochemistry at chirality centers, particularly in carbohydrate chemistry. A tetrahedral carbon is replaced by a Fischer projection by two crossed lines. The horizontal line represents bond coming out of the page, and the vertical line represents bond going into the page. For example, R glyceraldehyde, the simplest monosaccharide, can be drawn as shown in figure 25.1. Because a given chiral molecule can be drawn in many ways, it's something necessary to compare two projections to see if these represent the same or different enantiomers. To test for identity, Fisher projection can be moved around on the paper, but only two kind of motions are allowed. Moving a Fisher projection in any other way inverts its meaning. Fisher projection can be rotated on the page by 180 degree but not by 90 degree or 270 degree. Only a 180-degree rotation maintains the Fisher convention by keeping the same substitute groups going into and coming out of the plane. In the following Fisher projection of R glyceraldehyde, for example, the H or OH groups came out of the plane both before and after a 180-degree rotation. A 90-degree rotation breaks the Fisher convention by exchanging the groups that go into the plane and those that come out in the following Fisher projections of R-glyceraldehyde. The H and OH group come out of the plane before rotating but go into the plane after a 90-degree rotation. As a result, the rotated projection represents S-glyceraldehyde. A Fisher projection can have two groups held steady while the other three rotate in either a clockwise or a counterclockwise direction. The effect is simply to rotate around a single bond, which does not change the stereochemistry. RS stereochemical destinations. RS stereochemical designations. Section 5.5 can be assigned by the chirality centers if in a Fisher projection by following three steps as shown in worked example 25.1. Step 1. Rank the four substitute in the usual way. Section 5.5. Place the group of lowest ranking, usually H, on the top of the Fisher projection by using one of the allowed motions. This means that the lowest ranked group is oriented back away from the viewer as requested for assigning configuration. Step 3. Determine the direction of rotation 1, 2, 3 of the remaining three groups and assign R and R or S configuration. Carbohydrates with more than one chirality center are shown in Fisher projections by staking the center on the top of one another with the carbonyl carbon at or near the top. Glucose, for example, has four chirality centers staked on top of one another in a Fisher projection. Such representation doesn't, however, give an accurate picture of a molecular's true three-dimension conformation, which is curled around on itself like a bracket.